triponucleozygotes, but basically just about an attempt to genetically engineer human embryos. So what the researchers in this paper did basically is they tried to, as I said, genetically engineer some embryos using this system called CRISPR-Cas9, which we'll talk about in just a second, in an attempt to create what are called designer babies. And they're trying to identify problems with using this system, in the future that we're going to have to solve if we want to create designer babies. Now this term designer babies, I just mentioned it, Sydney mentioned it. If you haven't heard that term before, it's basically just a term used to describe genetically engineered babies that have been altered to give them a more favorable traits or something like that. And really, up until now, designer babies have only existed in the realm of science fiction. But this paper sort of represents the first step towards that science fiction becoming like a reality. So the system CRISPR-Cas9 that I mentioned, the way it works, there's two main components. The CRISPR protein, right, this green thing here, and the guide RNA, or gRNA, right here. So the guide RNA has a very specific nucleotide sequence that corresponds to a sequence somewhere in the DNA, like right here. And it'll actually bind to that sequence in the host genome. So once the guide RNA has found that sequence and has bound to that sequence, like right here, the Cas9 protein will actually cut the DNA, both strands of DNA, and you'll get a cut looking like this. So this is important because by changing the sequence of nucleotides in the guide RNA, the researchers can actually choose specifically where in the genome Cas9 is going to cut. So now once, cut piece of DNA is a problem for the cell. So once the cell identifies that there's been a cut made, it's going to try to repair it. And there's two pathways to repair, either HDR or NHEJ. So NHEJ stands for non-homologous end joining. Basically all that means is the cell takes the two cut ends of DNA and puts them right back together. The problem with this, though, is that while those two pieces of DNA are cut, you can have either nucleotides binding on or other extra nucleotides falling off. And so you might end up with some mutations, which could be harmful to the cell and potentially dangerous. So instead, what the cell can do is called HDR, or homologous recombination directed repair. And what that means is the cell finds a similar gene or an extra copy of the gene and uses it as a template to reconstruct the cut area and identify if there's been any mutations made. So basically, when the researchers are trying to introduce a specific, either a new sequence, a new gene, something like that, what they do is they inject the cell with an extra copy of the gene, of the cut gene, with that mutation already in it. And so that way the cell will use HDR to repair the cut using that extra copy of the gene as a template, and so that the copy now in the genome will have the same mutations as a template. All right, does everyone understand this CRISPR-Cas9 homologous recombination stuff? All right. So the first thing the researchers had to do was choose the guide RNA that they wanted to use in the actual embryos. They had three potential guide RNAs, G1, G2, and G3. And to choose between them, they didn't use embryo cells because those are kind of hard to work with and hard to culture. Instead, they use these HEK293T cells, which is just a very commonly used cell line that's easy to culture and easy to grow. The gene that they're going to be looking at in all of these experiments is called HBB, or human beta globin, which is just a component of hemoglobin. And there's another gene I'm going to mention called HBD, it's related to HBB. It has a very similar sequence. And because of that, the cell can use HBD as a template for homologous recombination directed repair. So these graphs, basically all you need to know about them is that a single solid line means that there's no cut made, so there's no gene editing done, basically. And if you see multiple fainter lines, that means that there is a cut made, so there is gene editing done. So just looking at this top graph right here, can anyone tell me which of the three guide RNAs look like they actually successfully cut gene? Anyone?
1 and 2?
Yeah, 1 and 2, exactly. So guide G1 and G2 were actually able to modify the gene. G3, as you can see, was not. So they kind of just dropped that one. And then to choose between G1 and G2, they looked to see how specific they were. So, they don't want these guide RNAs to be binding to any sites other than the ones that they're specifically trying to modify. So what they did is they chose 7 off-target sites labeled 1 through 7 that had very similar sequences to the one that they're trying to edit, to look to see if either G1 or G2 would accidentally cause mutations at those sites.
So if they're going to be editing sequences in the future, will they have to go through this whole process of like being able to specify like a certain type of guide RNA?
They're going to have to identify, specific. They're going to have to design guide RNAs that will only bind to that sequence.
This is your second presentation, right?
Yeah.
Okay. So the G1 and G2, that's the same exact G1 and G2 from the ones that modified the HBB gene, right? The ones on the bottom, which are looking at the similar sequences to HBB?
Yep.
Okay. And do you know how similar the 2 sequences on the bottom were to HBB?
Well, they're looking at 7 sequences, first of all. And I don't know offhand exactly how similar they are. So as you can see, looking at these 2 graphs, neither G1 or G2, for the most part, neither of them cause very many mutations outside of HBV. But it's kind of hard to see. There's a little bit of shading here at site 4 for G2, which indicates that there was a little bit of modification done there. So they decided just to continue with G1 for that reason. So now they moved on to testing with actual human embryos. Now the embryos they're using come from what are called 3PN, or triponuclear zygotes. And so triponuclear zygotes, basically what happens when an egg cell is fertilized by 2 sperm cells instead of just 1. So it ends up with an extra copy of its DNA. So if you look in this image, you can kind of see the 3 different nuclei right here in the middle. And so these cells, they can live, they can reproduce, they can split and stuff like that, but they can't differentiate into specialized tissue groups or organs or anything like that, which is sort of important from an ethical standpoint, because it means that there's no way that these cells could possibly develop into a full human. So they weren't really testing it on actual human embryos, just genetically similar embryos. So they did the CRISPR-Cas9 thing again on HBV, and they started with 86 embryos. Of those 86, 28 of them were actually cut by Cas9. But of those 28, only 4 were edited with the piece of DNA that the researchers injected. So that means only 4 of the 28 Cas9 cleaved embryos actually acquired the mutations that the researchers are trying to introduce. Another 7 of them were edited using HBD, which is that similar related gene I mentioned earlier, which is a problem because it means, again, they're not acquiring specific mutations the researchers are trying to introduce. And then the remaining 17 Cas9 cleaved embryos were actually repaired using that non-homologous end joining thing I mentioned earlier, the cell just sticks the two pieces of DNA back together, which, as I mentioned, is a problem because that can cause mutations for one thing, and also, again, it means that they're not acquiring the specific mutations that these four are. So this is sort of the first problem they identified, is that the cells weren't always using the DNA that the researchers want them to use. The second problem they identified, they looked at how specific they were, how specific this Cas9 system was. And so they looked at these 7 sites that they're mentioning here, are the same 7 that they talked about, here. And so what they're looking at is basically whether or not this is causing mutations at those sites. Again, same thing as they were looking at before, except this time they're actually sequencing so they could be more specific. And what they found is that actually in a few of the cases at these 2 sites, G1OT4 and G1OT5, you were getting mutations. Now this is again a problem because they did not intend to create mutations at those 2 sites. And there's always the possibility that these mutations are harmful, to the cell. So that's sort of another problem that will have to be addressed. So, just to summarize all the data so far, it is possible to edit human embryos using this CRISPR-Cas9 system, but there are some problems that have to be addressed. First of all, is that the cells had trouble acquiring the specific mutations that the researchers were trying to introduce. They didn't always repair their DNA using the DNA that the researchers gave them. And the other problem is that Cas9 has some specificity problems, where it'll cause mutations in sites that the researchers did not intend, which could potentially be harmful to the cell. So does everyone sort of get all the scientific data stuff?
Can you go back to the previous slide? What's the red and what's the green there?
So red basically just represents mutations. So that's different than the original. These are nucleotides that are different than the original sequence that was there. And the green is part of the CRISPR-Cas9 system that I was talking about. It's called the PAM sequence, and basically that's a sequence that the Cas9 actually binds onto and cuts.
Okay. I just, I don't know the system that well, so I'm
Yeah.
just trying to figure out. Why are the CRISPR system, the CRISPR tags on the end, I guess, why are those all different? Not all different. Why?
There are multiple different sequences that I think CRISPR, the Cas9 protein will bind to.
Do you know how long? So do you know how long the sequence is? How long the sequence is necessary for? How long the target of the CRISPR-Cas9 system must be?
So the sequence that the guide RNA binds to is, I think, only about 20 nucleotides long, and then there's this additional 3 nucleotide long PAM sequence that the Cas9 protein binds onto. All right. So if everyone gets a sign up.
I should have one question. Is there any advantages or disadvantages when I'm talking about this CRISPR-Cas9 editing and sequencing in having 2 sperm?
That was just it was sort of an ethical thing so that the embryos don't actually develop into actual humans.
But could it cause like any?
They didn't mention. I don't think so because each individual copy of the chromosome are still the same, but I don't know.
So you said that the eggs that are like double spermed, do kind of develop to a certain point. Do you know what point they develop to?
I think they can reproduce, but they can't differentiate. So they're all going to be regular, zygotes. Alright.
So is the guide RNA so if they found a successful guide RNA, is that specific to just one person or does that work for everyone?
It would depend, I think, on the specific gene you're trying to modify.
So
I think.
Like G1 could be specific to, say, if you want your baby to have blue eyes, but then G2 could work if you want it to have like blonde hair.
No, so the guide RNA is for a sequence that's already in their genome. So you would probably have to look at the sequence that's already there and create a guide RNA that's complementary to that, and then the additional piece of DNA that you inject would then correspond to the gene you're trying to create, basically, the trait you're trying to create. So, aside from all the scientific and technical problems, there are some interesting, ethical questions that are raised because, researchers are starting to look into this. So, like, when this paper was released, it caused a lot of controversy in the news because people feel that this sort of line of research is kind of unethical, which I think is in part because people are just afraid of new things that they don't really understand. But it's also, there are some legitimate ethical concerns that people have. So some things just to think about and maybe discuss is how much control should parents actually have over their children's genome. So like for instance, I think if someone wanted to use this technology to say fix a mutation and prevent their child from suffering from a genetic disease, I think most people would agree that that's okay. But what if you wanted to edit something less essential? What if you wanted to improve your child's strength or speed or intelligence, memory, or even something purely cosmetic like eye color or hair color? Should there be a line between what is actually important to control and things that are just so trivial that we shouldn't really have any control over them? Also, there's a question of, if this technology becomes more commonplace, what will it mean for people who say can't afford to have this done or don't have access to the necessary facilities? Or even people like us who are born before this was really a possibility. Would we become, sort of second-class, inferior citizens? Or would we have to impose some sort of restrictions on it? So if anyone has any thoughts about these or any other ethical things, feel free to talk.
If everyone were creating a design baby, I assume that you would pick almost every trait to make your child as perfect as possible. Would that affect the diversity of the gene pool if there's a bunch of perfect people running around that know? I mean, I assume they at some point have similar genomes.
I think that's definitely a problem. That could be a potential problem.
And isn't it, going off of what Chase said, isn't it also kind of messing up with the natural evolutionary process by going in there and affecting the DNA and the mutations that wouldn't normally be taking place due to like the evolutionary processes?
Yeah, I suppose.
What if this is a mechanism of evolution? What if, like, part of evolution is designed to generate a intelligent human being capable of non-homologous end joining, editing, gene sequences? Like, what if this is part of it's just a piece of our further evolution?
Would we not be evolving? Everyone would be evolving at such a different rate because of this status of like wealth. Like we would have such a large population that would be able to evolve, and they would be at a disadvantage. It would be inferior if we created these perfect children.
I mean, certainly, if this, again, we're making a lot of assumptions here, but it's a good question. But yeah, if wealth allows you to do CRISPR-Cas9, and you're able to do this great number of times to a human genome, then yeah, you would theoretically be influencing the rate of mutation in one group of people more than another.
It may just be me, but this doesn't concern me all that much. I mean, potentially the strength of these genetic modifications could be significant, but they've done a few studies, particularly studying the correlation between IQ and long-term success, and there's almost no correlation. Having a higher level of IQ doesn't actually guarantee longer-term success than having a good life. So I think this is kind of ignoring some of not just the epigenetic factors, but also the social factors that lead to people's ability to engage with society.
Although I think there are also genetic factors involved in things like confidence or creativity, which will have a lot, a much bigger effect, I think, on someone's success. So I think it is possible that you could design a child that's more successful.
Last one.
I'm not a scientist, I'm an English teacher. I see the good side of this is this leads to the eradication of genetic diseases like cystic fibrosis and spina bifida. The dark side of this is that it leads to Aldous Huxley's Brave New World.
What is Aldous Huxley's Brave New World for the science people?
Genetically designed society, everyone perfectly genetically engineered to suit certain purposes and functions in a perfect world.
What was the downside of it?
Genetically designed world is that, again, where the society is simply engineering, designing, and producing workers, drones, brilliant people, creative people, according to a genetic formula.
I'm sorry, I don't understand. What's the
Free will.
downside?
There you go.
Joking, sorry.
You could design people to just be like this.
I say we try it.
Next week, Will Evenson and lots of breakfast. Hope to see you all there.
Breakfast!